Hello, everyone, and welcome to today's webinar, Proven Tips and Tricks for Ensuring the Success of ELISA Experiments. I'm Christy Jewell of Labroots, and I'll be your moderator for today's event. Today's educational webcast is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, we encourage you to participate today by submitting as many questions as you want at any time you want during the presentation. We'll answer as many of your questions as we have time for at the end of the presentation. To submit a question, simply type them into the Ask a Question box and click Send. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. Today's presentation is educational and thus offers free continuing education credits. Please find the Continuing Education tab on the far left hand of your screen and follow the link to obtain your credits. And with that, I'd now like to welcome our speaker, Ricarda Heintz, Manager, Research and Development Biosciences, Thermo Fisher Scientific. For a complete biography on our speaker, please see the Presenter tab at the bottom right hand of your screen. Ricarda, welcome. You may now begin your presentation.
Thank you, Christy, for the introduction. So just let us go through to the agenda very shortly. First, we will start with a short introduction to the Vienna site, and then we will jump right into the topic of ELISAs, talking about best practices, assay development, and products. And we will end with a short section on troubleshooting and customer services. The Vienna site was established as the site of Center of Excellence for Immunoassay Business in 2017, with over 25 years of experience in immunoassay development. The site is dedicated to development, production, and quality control, and marketing in order to support worldwide sales of immunological research reagents and research diagnostics. All of our products are branded with the Invitrogen brand. Well, now let's talk about ELISAs. ELISA was first described in a paper in 1971, and since then it became the gold standard for quantitative analysis of proteins. It has a lot of advantages, for example, short turnaround time. It only requires basic lab equipment, it is easy to automate, and a very reliable and cost-effective tool. Due to this versatile and advantageous aspects, it's applicable in many different areas like diagnosis, monitoring, allergies, drug development, quality control, and of course, research. So there are different types of ELISAs. The most basic form is the direct ELISA, where you coat an antigen to the plate and detect it with a primary antibody that is conjugated to an enzyme. This assay is quick and easy, but it can only be used for highly abundant targets because it lacks sensitivity and specificity. The indirect assay is an already improved format of the direct assay. You have also an antigen coated to the plate, but you use two different antibody to detect this antigen. So it's more sensitive and more versatile than the direct ELISA. But the most common form and broadly used type is the sandwich ELISA, where you have a capture antibody on your plate, detecting your antigen, and then a detection antibody comes in, which can be a primary antibody or a secondary antibody, again labeled with an enzyme. This assay is highly sensitive and specific, but it also needs a lot of optimization to identify the right antibody pair. And last but not least, there's also the competitive ELISA, which can be used for small proteins because it's very sensitive, but it often lacks a certain type of specificity. These competitive ELISAs detect your antigen with a capture antibody, but this antigen competes with a different antigen that is conjugated to the enzyme. So your signal will be indirectly proportional to the amount of antigen in your sample. Now let's talk a little bit more about sandwich ELISAs because it is the most common form of ELISAs. As said, it usually starts with an antibody coated to the plate, and you incubate this antibody with your set standard or sample, which is followed by a washing step to get rid of any unbound antigen. After that, you introduce a detection antibody, building that, let's call it sandwich, and you have different options there. So you can go either with a primary antibody or with a secondary antibody, and they can be labeled with biotin or HRP, depending on your assay setup. After your HRP conjugate or biotin conjugate, you again will have a washing step and add your substrate, which is in most cases TMB. After a short incubation, you have your stop solution ready and the whole assay can be read in your ELISA reader. Now let's talk about a little bit about best practices in your ELISA experiment. We always recommend to plan your experiment in advance. So read the manual carefully and make your plate layout, planning everything you want to get on the plate. Always include blanks and controls, and we recommend to run samples and standards in duplicates. If your experiment requires triplicates, go ahead and do so, but the minimum should be duplicates. Always calculate your sample dilution and reagent volume. Your manual will give you certain hints how to do it correctly. And you should prepare your standard curve according to manual and do not extend it in the higher or lower ranges because the assay is validated for the range that is stated in the manual. Make sure that you minimize the freeze-thaw cycle of your samples and reagents. And if the assay requires to dissolve a lyophilus standard, please do not freeze and reuse this standard after you used it once. In general, it is important to not mix reagents from different kit lots. It will not give you valid results, and therefore you should avoid it. Also make sure that before you dive into your experiment, that your ELISA reagents are on room temperature. Now let's talk a little bit about pipetting techniques. Use the correct pipette that is validated for the range you want to use it. And if you have available, use a multi-channel pipette. During pipetting, confirm that the tip is firmly seated on the pipette and that there are no air bubbles present. Always change your tips between each standard, sample, or reagent. As well, always change your reagent reservoirs. It's good practice to angle your pipette and do not scratch the bottom of the plate because there's your capture antibody, and by scratching the bottom of the plate, you might detach it. Another very important hint here is to maintain the same pipetting order when applying reagents. So go from left to right or from up to down, whatever you prefer, but maintain always the same order, especially when you pipette TMB and stop solution, because there it's most time critical. Incorrect washing is one of the main causes for poor assay performance. So really make sure that you have your washing procedure set up. Always completely aspirate the liquid from your ELISA wells, and also here take care to not scratch the inside of the well. Fill the wells of the plate to the brink, so at least 400 microliters. and it often requires to sit for 15 to 30 seconds in the well. After washing, invert the plate and tap dry on absorbent tissue. Don't be hesitant about it. There should be really no residual liquid left in the well. Alternatively, you can also use a squirt bottle or an automated plate washer, which will increase the reliability of your experiment. Incubation time and temperature is also critical for your experiment. So follow the manual instructions and make sure that the temperature in your lab is consistent across each incubation step and different days. Always consider assay kinetics, so do not let your plate sit dry during your ELISA experiment and immediately add reagent after each washing step. Also make sure that you apply standards and samples at the same time. So make sure in advance that your samples are thawed so that you don't have any unnecessary pauses between applying your standard and your sample. Shaking can be also pretty critical to get reliable results. Some assays may need mandatory shaking, but also make sure that you use the right shaker type. So in most cases, this will be an orbital shaker, but also make sure that you use the right speed and that the shaker has the correct radius. Always make sure that you do not stack your plates during incubation. This might lead to edge effect, which I will explain a little bit later. And use a new plate sealer for each incubation step. Do not reuse them. Now let's talk a little bit about your analysis. So it is really recommended to run a standard curve on every plate. Do not reuse any formally generated standard curve data and use a curve fitting software, which often comes with the ELISA reader, or there are free versions available to use, but in any case, use a 4PL or 5PL fit. Then calculate your sample concentrations based on the fitted standard curve. Use the average OD values and subtract the blank OD from your sample and standard OD. Do not forget to include your sample dilution factor. And in general, we recommend to calculate the coefficient of variation, which is standard deviation divided by mean times 100%. And your CV shouldn't be higher than 20% for your standard and sample. In case it is higher, there might have gone something wrong with your experiment. Additionally, if samples fall outside the range of your standard curve, make sure to reanalyze them with a higher sample dilution in a later experiment. Well, we already talked about a lot of factors that affect your signal generation ELISA, but there are many more to consider. For example, your assay plate, the material you use, the well shape, or if it has a treated or untreated surface. Also, the usage of buffers, especially coating and blocking buffers, their composition and pH value can have an effect on your ELISA. And of course, the most critical points like antibodies, specificity, the concentration, or the affinity can lead to different results in your ELISA experiment, as well as incubation time and temperature. There are differences in enzyme conjugates you can use. I already talked a little bit about biotin and HRP, so there are certain types you can use, but they might also have different activities, or you might need to use a different concentration. You always need to consider the antigen, your target antigen as well, the conformity, the stability, and its available epitopes can have an influence on your ELISA experiment. We already talked a lot about washers. So the composition of your wash buffer, the volume you use, the duration and frequency are also pretty important. Substrate can also lead to different sensitivities of your assay. So make sure that you choose the right one, and it might be important to consider the age of your substrate, or if it has been pre-exposed to light. And last but not least, there is also the type of ELISA reader that you use, which might use different filters or different exposure times than other types. So overall, there are a lot of things to consider for your ELISA experiment. And there are certain advantages to have an ELISA kit or to build your ELISA kit yourself in your lab. The development process can be quite tiring. It usually starts with an antibody pairing, which we usually recommend to start with at least 6 different antibodies. So you will have 36 pairs to test. And it also is recommended to use 2 different concentrations of detection antibody. And sometimes we even suggest to use 2 different proteins to make sure you choose the correct one. After your pairing, you usually go on with the evaluation of your standard range, LLOQ, ULOQ, and sensitivity. Next step is to check for specificity. So does your assay really detect your target antigen? Or in case it does cross-react with any other proteins. So make sure you all have that done. And after all that, you just have your final antibody pair selected, but within a very cost and labor-intensive process. And after that, you do not even have a ready, robust, and consistent assay kit. So here I will shortly talk about our in-house procedure. It usually takes us 6 to 9 months of development work to find the best suited antibodies and proteins. We always check for optimal assay design. So is it a sequential or a batch incubation? And it is always important for us to find a short turnaround time. After that, we always verify the sample matrix and dilution. So we do spike recovery and dilution linearity to make sure that we do not see any matrix effects. And we always recommend the correct sample dilution. After all that, we start with assay robustness testing. So we check for the correct incubation time, temperature, and assay kinetics, and do a reproducibility and stability testing of our assays. We also make sure that we have all the components stable formulated in the kit. And then we go into validation, where we determine inter-assay CV and intra-assay CV. Also, our quality control makes sure that our assays do fulfill a lot-to-lot consistency, so that you will have a reliable and reproducible product. Talking about our product, here we have a short overview. So we have our uncoated ELISAs, where you will get all the reagents to build up your own ELISA in your lab. But we also have our coated ELISAs, which are our highly validated gold standards ready to use ELISA kits. But we also offer instant ELISAs, which are very streamlined ELISAs that will maximize your accuracy, reproducibility, and it's a very time-saving method. And we offer also 2 different products. One is a PCR system-based PLA assay, the ProQuantum immunoassay, and we also offer products for multiplexed protein or gene detection, which are called ProCardaplex and QuantiGeneplex. At Thermo Fisher, we have a high variety of ELISAs spanning across 2,000 different targets to choose from, different species like human, mouse, rat, and many more. And they all come with the recommendation of the correct samples to use and the correct dilution to use. Now I just wanted to give another short overview about the instant ELISA kits, because it's really a way to make your ELISA experiment more effective and time-saving. With the instant ELISA kit, you will have everything readily lyophilized on the plate. You just need to put on your sample, incubate the assay, wash it, add TMB, and then add your stop solution. It's a very quick and easy way to do your experiment in case you don't have time to set up a complete assay. Now we will talk a little bit about troubleshooting. So I brought some experiments, some results from our lab for you to have a look. So on the left side, we have a beautiful standard curve with high assay signal and a low blank signal. But on the right side, when you compare it to the left side, you will see that we here lack signal. So we have a low standard, and our last 3 standard points are almost the same value as our blank. So what might have happened? Possible causes are that the reagents we used weren't at room temperature, or that we stored the components at an incorrect condition. Make sure that you check the manual at which temperatures they need to be stored. The reagents might have been expired, or we prepared them incorrectly. But it might also been the case that there wasn't enough detection antibody, or that the walls of the plates were scratched with the pipette. Also make sure that the plate reader is set at the correct wavelength. This can also have an influence on poor or weak assay signal. But what happens when the assay signal is too high? So again, you have our beautiful standard curve on the left side. And then when we have a look at the right side, you will see that the first 3 standard points do have almost the same OD value, and that there is a pretty big gap between our last standard point and the blank. This might be due to insufficient washing, or that the reagents or the standard dilution was incorrect, or that we had any issues with the dilution. Also, it might have been the case that we incubated the assay longer than intended, and this might also lead to a high signal in your ELISA. So now we are seeing a very high background signal here. So when you compare the blank value on the right side to the blank value on the left side, you will see that it is pretty increased, and that the last 3 standard points do almost have the same value as the blank. This might also be due to insufficient washing, or maybe that the substrate which was used was exposed to light. But it might also have been the case that the assay was incubated for too long, or that there was a mistake in the standard curve preparation. So let's talk about reproducibility. In case you experience any poor CVs during your ELISA experiment, it might be due to any residual liquid in your wells after washing. So really make sure that you tap your plate on a paper towel after each washing step to get rid of any residual liquid. It might also be necessary to increase the duration of your soak steps during washing, or that you might have used the wrong coating or blocking procedure for your assay. Also make sure that you do not reuse any plate sealers during incubation, but poor CVs can also come from any pipetting errors. In case you see any variability from day to day during your ELISA experiment, it might be due to inconsistent incubation temperature in your lab, or that you even use a completely wrong incubation time for your assay. So make sure that the temperature and the time are set correctly. Also, edge effects can appear in your ELISA experiment. Edge effects is usually when you see higher signal at the outer walls of your plate than on the inner walls of your plate. So the ones closer to the frame give you a higher signal than the ones far away. This happens when you stack your plates during incubation or if you insufficiently sealed your plate. So make sure that the plate sealer is set up and you don't stack your plates. Also, in case you use any incubator and your incubation temperature is at 37 degrees, make sure that the air circulation in your incubator is consistent and that your plate is placed in the middle of your incubator. So now I will just very briefly talk about our customer services. So in case you're looking for an ELISA and you don't want to do it yourself and you want an experienced team working on it, we also do custom assay developments. We are operating on ISO 9001 and you will receive regular development updates from us. And in any case, you have your samples ready in your lab, but you can't find any time to measure them, feel free to reach out to us. We will do the sample measurement for you in all of our assays. And with that, thank you very much for your attention. I hope you enjoyed your webinar. And now I will open it up for any questions.
Thank you, Ricarda, for that informative presentation. It is now time to begin our live Q&A portion of the webinar. Now to our audience, if you have a question you would like to ask, please do so now. Just click on that Ask a Question box located at the bottom of your screen, and we will answer as many of your questions that we have time for. Okay, let's get started. We have some great questions already coming in. Ricarda, let's start with this one. Please describe LLOQ and ULOQ.
Thank you very much. This is an excellent question. So LLOQ stands for lower limit of quantification and ULOQ stands for upper limit of quantification. So this is basically your highest and your lowest standard point of your standard curve.
Very good, thank you. Our next question. How do you detect a limit of detection in a competitive ELISA?
Well, actually, this is very similar to a sandwich ELISA. We have a recombinant protein that is biotinylated, and we have a recombinant protein that is not biotinylated. So when you add both of them into your assay and you perform a dilution linearity of your biotinylated antigen, the last concentration, which is still distinguishable from your blank, this is your limit of detection here.
Very good, thank you. Next question. When using the automatic washer, is tapping between washes necessary?
When you mean like you do one wash and this is 4 washes, then no. But at the end of your wash cycle, always tap your plate on a paper towel.
Next question. Could you please elaborate on edge effect? Is any stacking of plates permissible?
I would say the only time you are allowed to stack plates is when you maybe do coating and blocking. Or when you have an incubation at really low temperature. So for example, you have an incubation at 4 degrees Celsius. Then it's usually okay to stack the plates. In general, every incubation that is on room temperature or on any higher temperature, I would in generally avoid it.
Very good. Ricarda, poly or monoclonal antibodies, what should be used for an ELISA?
A great question. And this question is always asked. So in general, of course, it would be good to use monoclonal antibodies. With monoclonal antibodies, you will ensure that even if you bring up in a different lot of the antibody, you will still get consistent results. This might not be the fact when you bring in different lots of polyclonal antibody. But I know it's difficult to always get two monoclonal antibodies for your ELISA. Sometimes they are not available. If so, it is a good idea to have the monoclonal antibody as a capture antibody. So you also already have a very specific capturing of your antigen, and then use the polyclonal antibody as your detection antibody.
Thank you. We have some great questions coming in. Let's go with this next one. How can I distinguish between a weak signal or a minor case of unspecific antibody binding?
Well, we usually do, or you should do a specificity test. So you should really make sure that your ELISA is specific before you go into large-scale sample screening. So what we normally do is we coat an unspecific antibody to the plate and add the samples and do the same with a specific coating antibody. Then we add our detection antibody, and then we double check what signal do we get on the plate with the unspecific coating antibody and what signal do we get on the plate with the specific coating antibody. You should really see no signal on the plate with the unspecific coating antibody. Only then your assay is specific and you should go into large-scale sample screening.
Very good, Ricarda. Thank you. How can you guarantee that the results are not unspecific?
Well, it's mainly the same as I answered right now. So you really need to perform this experiment with the unspecific antibody and the specific antibody to make sure that you only have specific signal in your assay.
Okay, very good. Next question. This one is two parts. Would it be possible to use sandwich ELISA to determine the concentration of a recombinant protein present in cell culture media? And is it necessary to use cell culture media without phenol red?
Well, I would say both cases, yes. Of course, you can detect a recombinant protein in a cell culture medium. And you can also use a cell culture medium without phenol red. This shouldn't be an issue at all.
Okay, very good. Our next question, how long does it take to develop an ELISA in general?
Well, if you perform all the steps I have described, it really takes approximately half a year, so 6 months. So we usually have a feasibility phase where we go into pairing, establishing our standard curve, looking for specificity or for any cross reactivities. This usually already takes us 3 months. Then we invest another 2 months where we do optimization, where we look at matrix effects, do spike recovery experiment, dilution linearity experiments, run day-to-day reproducibility. And And of course, stability testing. Stability testing is always a very important factor when developing ELISA, because you want to make sure, of course, that you get the same results from your ELISA over a period of time. And after that, we usually go into validation and into like also going into user-to-user differences. This takes us another month or so. So in total, it's 6 months in total. This is usually the time we calculate for an ELISA development.
Thank you, Ricarda, for this very informative Q&A session. Do you have any final comments for our audience?
Well, I really hope that you enjoyed this webinar. I really hope that you will have now some tips and tricks how you can improve your ELISA experiments. And in case you have any more questions, or you are interested in any of our customer services, feel free to reach out to us.
Thank you again, Ricarda, for your time today and for your important research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Now, before we go, we do want to thank our audience for joining us today and for their interesting questions. There were some questions we didn't have time to answer today, and also those that are submitted during the on-demand period. They will be addressed by our speaker via the contact information you provided during the time of registration. This webcast can be viewed on demand, and Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, take care and have a great day.